Thank you, everyone. Thank you for the feedback. I will quickly go through some points just so we can have a seamless virtual conference experience. In order not to disturb the courses of the conference, we request all of you to stay on mute during the lectures and use the chat box to communicate in exceptional cases. You may unmute yourselves during the question and answer segment of the lecture to interact with the speakers. Breaks are planned through the course of the day. In case you have any technical issues, please signal me in the chat box and we will attempt to resolve the issues promptly. I'm happy to note that this conference is also live on YouTube right now, and we'll be sharing the link in the chat so that you can share it with your friends. We also have some exciting quiz competitions at the end of each day, and the questions will be taken out of the lectures that are presented on each day. So please make sure to listen attentively to all the lectures. And winners will also receive exciting prizes. As mentioned earlier, this conference is being hosted by T-Sweries in association with Dare to Start and Pharma Academy. And I'm pleased to inform you all that all the key personnel for launching this conference are here with us today and will be sharing brief notes of welcome. To kickstart the inaugural segment of the conference, may I invite Dr. Praveen Mamadala, Joint Secretary, Telangana Social Welfare, to present the welcome address. Dr. Praveen Mamadala is the Joint Secretary for Higher Education and the Department of Alumni Relations, Telangana Social Welfare Residential Educational Institution Society. He has over 22 years of teaching experience across various universities. He is an associate professor and the head of the Department of Biotechnology at the Telangana University and specializes in plant biotechnology. He has authored high-impact publications even while pursuing his doctoral and postdoctoral degrees from Shanghai and The Ohio State University in USA, respectively. Dr. Praveen is a powerful force in the workplace and uses his positive attitude and tireless energy to encourage all of us to work harder. He is also responsible for truly manifesting the Social Welfare Residential Degree College's motto, Opportunities Unlimited, by developing multiple MOUs with premier institutions and establishing flagship initiatives that have created avenues for student and faculty development. Sir, may I invite you to present the welcome address.
Thank you very much, Shiva, for your brief introduction about me. I don't know whether I deserve it or not, but it's all because of you people and all the officers in the head office. Okay. So, good morning, one and all. It gives me really an immense pleasure to welcome you all at the DNA at the rate 150 conference. My special thanks goes to my co-convener, my colleague, Dr. Pavani. Without her, I think it's not possible for me to do such conferences. And also equally, a powerful young guy, Dr. Jagdish Ganda, from Dare to Start, and also a senior most professor, Pandu Reddanagaru. Without him, without his advisors, without their suggestion, it is an impossible task for me to organize such a wonderful event. Thanks to all. And top on this, my special thanks goes to Sri D. Ronald Rose, IAS officer. Again, he's a very young guy, like Jagdish, very young, vibrant, dynamic. I don't know what to add all, but he's a multitasker. And when I told him about this conference, he simply told Praveen, go ahead, it's a wonderful conference. But because of some important work at Secretariat with Chief Secretary and with the Principal Chief Secretary of the Education Department, he may join or he may not join, but he conveyed best wishes to one and all. So, as you are already aware, we are here to celebrate the greatest discovery, I can say, 150 years of DNA discovery. And while you will be hearing many interesting lectures on the advancements made in the science following this marvelous discovery, the teacher in me is really excited because I'm a teacher. I'm not at all an administrator. To give you a brief background on set expectations for today and also tomorrow's sessions. For those of you, particularly from school students, I can see the school students from Amangal and school students from Adilabad and Piknoor, different places students have joined. So for them, Frederick Mishra, who first laid the foundation about the DNA discovery way back in 1869, and took almost 2 years to publish. Actually, I should say DNA discovery is past 150 years, because if you calculate back to 1869, but it was published after 2 years in 1871, if I'm not wrong. And it was until again 1940s, long gap. No one knows about the DNA's role in the genetic inheritance, and till date, it's a mystery with the DNA. Whatever you do, you will. It's a mystery. That is the reason why yesterday I was me and Dr. Pavani, we were going through online, like how many Nobel Prizes we got in the DNA world. Just like that, within 2 minutes, when we searched the list of the Nobel laureates, around 20 Nobel Prizes are in the chemistry, like medicine and physiology, physics, related to the DNA. 20 within no time. I think it will be more than 20, of course, but the outstanding Nobel Prizes comes from this DNA discovery. So, my dear friends, I think you'll be having a wonderful day ahead, and tomorrow also. Today you will not sleep, I'm sure, because of some fun activities and some quiz competitions which are planned by our organizers. So enjoy the session. I once again welcome you all. I hope you all find this seminar highly engaging, fruitful, and beneficial for your future venture. I especially thank the principals who made the young students to come and sit and attend this wonderful seminar. It's exclusively dedicated for those students. Thank you very much. Over to Shiva. Dr. Pavani, if not
Shiva. Yeah. Thank you so much, sir, for welcoming the participants. Now, may I request our partner from...
Thank you, sir. Thank you so much for those energizing words of welcome. Today, we're also joined by Dr. Jagdish.
I think there's a lag.
Oh. Should I speak?
I think there was a technical issue. There's some lag, but yeah, Shiva, please continue.
Am I audible now?
Yes, Shiva, please go ahead. Yes, Shiva, please go ahead.
Okay, all right. Thank you so much. All right. As I was saying, today we're also joined by Dr. Jagdish Gandla, our organizing.
It's okay. It's my turn. Yeah. So, hi everyone. My name is Jagdish Gandla. I'm from Kurnool, Andhra Pradesh, but I'm so happy to support Telangana State as well. We are all united.
Partner for the event. May I request you to address the participants? I'm going to address this. Sorry.
Okay, Shiva. Very good. So, good morning, everyone. Good morning, dignitaries of the event. Today, I want to, I will talk anyway tomorrow, mostly about the career skills and everything. But today, what I want to share with all the participants here is that why are we doing this event? And also why we chose these topics and why we have this kind of activities. So I want to give a little bit background. So when Praveen sir and Dr. Pavani has reached out to me and asked like we should do a DNA conference, then we thought like DNA is a very big thing. I mean, for when we look back, like as Praveen sir said, 150 years back, DNA was discovered. But within this 150 years, a lot has happened. From DNA, we discovered central dogma, like from DNA, mRNA comes, mRNA to protein. So there's a lot. Even you have seen mRNA vaccines from BioNTech, which we have for COVID. So we were not clear like what exactly is beneficial for students. So we have chosen a topic like from DNA, you will discover like how DNA has been exploited in omics. So we have very powerful speakers from all over the world. Tomorrow we'll talk more about DNA applications. So my request for everyone would be like, really be curious, like you come with a passion that, okay, I will learn something today. It's not like you get everything, but it's a journey. So at least for younger students, whoever are entering your career into the life sciences, make this as a first step, because the lectures, what you will discover today or tomorrow, may not be something that it may go into your brain immediately. But the thing is, these are the seeds that you plant in your brain so that this may help in your career. And some of the workshops. So this is also not a typical conference that you attend. Normally in the typical conferences, you will have mostly presentations, but here you are the main heroes. Like everything is around you. You please ask questions as much as possible. We are here to cater your needs. And as I said, like, be curious. You'll have poster presentations, you'll have scientific arts, and also you'll have DNA isolation workshop mostly tomorrow, because as Praveen sir said, the kits are stuck somewhere logistically. So we will address that as soon as possible. And you will isolate DNA and we will interact as much as possible. So yeah, over to you, Shiva. Many thanks for giving me this opportunity. Maybe Pawni ma'am, I think Shiva has issues.
Shiva?
Pavani, I think you can take over if Shiva has got some.
Yes, sir.
Thank you so much, sir. Is it any better now?
Yes, Shiva, please.
Yes, ma'am. Yes, please. Yeah.
Pawni, please go ahead.
Shiva, for some time, you please hold on. From hereafter, I think Dr. Pavani will take over. Over to Dr. Pavani.
Yes, sir. We are so sorry for the technical issues. But that's what makes the virtual events more fun. It gives you a reason to laugh. You'll remember this. If you don't remember the lecture, you'll remember the confusion that happened initially. But anyways, so we also have Professor P. Redana sir with us, who is an advisor for this event. He works, Professor P. Redana is a senior most professor at the School of Life Sciences, University of Hyderabad. Sir's research area is mostly the area of inflammation biology. He specifically works on cyclooxygenase and lipoxygenase pathways. Sir is actively involved in setting up of TBI and BioNest incubation centers and in promoting innovation and entrepreneurship activities on the campus. Sir is also the founder member of the Federation of Asian Biotech Associations, FABA, where he currently serves as the executive president. Recently, he launched FABA Academy to help students in their career development. Sir also served as the founder director for National Institute of Animal Biotechnology, which is an autonomous institute at the Department of Biotechnology, Government of India. So we are really delighted to have you with us, sir. So with this few words of introduction, I request you to welcome the participants. Over to you, sir.
So good morning. To you all, this unique 150 years of DNA discovery. I'm extremely happy that Telangana Social Welfare Residential Educational Institution Society has come forward to organize this event in association with Dare to Start, which was founded by Dr. Jagadish.
So we are losing your connection.
Sir?
Hello, Pavani.
Yes, sir.
I
think we lost Redana, sir.
Also, please check at your place, Pavani. There is kind of a resound coming. Just check once at your place. Yeah.
Yes, sir.
Just control the volume or whatever. Right. If Redana sir is lost connection, I think shall I go ahead to save some time? Because at 2 o'clock we have Rakesh Mishra sir. Am I right?
Yes, sir. I think you can go ahead, sir. So whenever Redanasar joins, maybe I can just share, like Redanasar will also, on behalf of Redanasar, tomorrow also we'll have a talk about Pharma Academy. So maybe you can go ahead now, sir, Praveen, sir.
Thank you very much. And before that, I request the Dr. Pavani or Shiva to follow up with Rakesh Mishra ji and see that you'll be joining by 2 o'clock with us. Okay. Okay, Pavani. Yeah, thank you very much. Thank you. So I'm back again, my dear friends. So once again, welcome to this wonderful event, international virtual conference. I should say conference and workshop, because since last 2 years, I can say, because of COVID pandemic, we are at our home and we stopped doing laboratories, right? Particularly undergrad students and postgrad students. So we thought, why not we should ship a kit so that they will do this exciting experiment with DNA isolation, which we almost forgot since last 20 years because of some reasons. Thanks to Chaitanya Narayana, I should say. I don't know whether I should say thanks to Chaitanya Narayana colleges. So at intermediate level, we stopped doing practicals. And the same was continued up to the UG, and in most of the cases, postgraduate colleges also, they're not able to do some practicals. So we thought, why not to have some exciting lectures today, combined with some DNA isolation kit with some other activities. So friends, coming back, we received really a great response. I even never imagined that a school student will register for an international conference. Generally, it will never happen, because when I was in my university, working in a university, even university teachers will never go attend for such conferences. But I have seen a school student, particularly from 5th class, 6th class, or 9th class, or whatever, they register for this program. This shows their a great curiosity. We always say that curiosity has kasi, particularly with my social welfare students. They have tons of kasi to do something, innovations in the social welfare. Thank you very much for teachers who are really motivating the students to join such spaces. And across the nation, we received a very good number. Actually, it was only 200 slot, but we received many requests. Again, we are open for the participation, and with which we got 50 more within no time. But only for the participants who registered later after deadline, they may not receive the kits, but they can attend the sessions very happily from their home or from their workplace. So come back to this today's conference. I should convey again thanks to Mr. Jagdish, who connected very big academic leaders or research leaders, international speakers from USA, Germany, Australia, and also from India. We have some very top-notch researchers joining this and educating us and empowering us also. So as I told you, the highlight of the conference is this do-it-yourself DNA kit. You may ask me, sir, what is this do-it-yourself DNA kit? Why it's called highlight? The only reason is if a student can see those fibers or the DNA whenever they're isolating, and definitely it will bring lots of aspiration. One is awareness and then it's aspiration. With that, definitely they will change their mindset, or they will think of what to do beyond this. So when I go back to my teaching or into the laboratories, till date an MSc student do not know how to isolate a DNA from bacteria, or they do not know how to isolate from a plant cell or animal cell. So they say, sir, some solution 1, solution 2, solution 3 we have to do, and they will go. They are done. They can't do anything. So this is just the beginning. And later in the next coming sessions also, we want to make it a regular event like this up to next year. 1 more year celebration. This is 1 year celebration. And we want to conduct several experiments which are very important for the genetic engineering or also genome editing. We will be having a wonderful lecture on the genome editing, because genome editing is such a great discovery, I can say. Tomorrow, I'm sure, after 10 years, no wonder you will be having a hospital by the suffix gene or genome. So they will start doing the treatment based on your genomic data. So such development we will see maybe by 2035 or max 2040 we will see, because already USA has started doing this. That's why we thought that our conference should not go, and we should have a mixture of lectures, quizzes, and very interesting poster presentations and art displays. Art displays is the idea of Dr. Pavani. I thought who will send us, but we received wonderful arts, very, very wonderful. And even the organizers tried, spend some time to understand what is that behind that art. So that's a beautiful art the students have made. And the date to start organization and Fava Academy, as I told you, the date to start it, because why I requested Dr. Gandla is the very important is biology people will never opt for entrepreneurship. I do not know why. The people biology or students from biology, they opt only two things. One is becoming a teacher or one is becoming a researcher. That's all. They feel entrepreneurship is a different world for them. But you will be having a very good lecture on what is the entrepreneurship and how to prove it. And we will definitely share lots of experiences on the Date to Start program. And also the Fava Academy, as Pandu Redana said, because of some technical reasons, he got disconnected, but for sure tomorrow will be joining us. And also we thought of having a platform to discuss various opportunities in the life sciences. Generally, we are not aware what are the opportunities. That's where we are gone. So when you join in a BPC, you will think that you want to become a doctor. That's all. If not doctor, you are an ordinary BSc. BSc होने के बाद you do BEd and become a teacher in majority of cases. Only few will think of going into Central University or some university. Again, after PG, unfortunately, I have many of my students who did BSc Biotechnology, PhD Biotechnology, and they will do BEd. Do you know why? They will get a job as a teacher. So they do not know the opportunities after PhD in biotechnology. That is unfortunate. That is happening till date. I am talking about 1996 ka story. 96 May, my senior did PhD in biotechnology. At that time, there is no biotechnology, but I still remember his name, but I don't want to tell his name. From a big lab, after PhD in biotechnology, he joined as a teacher. I don't say teacher job is a very small job or nothing, but when you have such a degree, you have to explore and see that you have to do lots of innovations, my dear friends. I will be sharing some of the ideas or generated by the Frederick Mishra later, how they did the experiment also. But if you do not know what you have to do, where you have to go, then it's based on doing science. So we'll be having a wonderful session on a brainstorming discussion to discuss on our career options in the life sciences. Coming back to this, I will take only 2 or 3 more minutes because DNA discovery is something it's like a great coincidence in the 1 decade time. If you go back to the history, just you can open Wikipedia and type 3 names. One is the great scientist Charles Darwin. I hope everyone might heard his name. Charles Darwin, a great evolutionary biologist. And I hope you also know Mendel. Mendel is, without this, we do not know what is this loss of inheritance. And other is Mishra. So these 3 people are in 3 different places. All these 3 people, okay? At that time, there is no Google, there is no phone. So they are in different places. They never got connected. But 3 have done marvelous discoveries. As Charles Darwin, the origin of species, natural selection theory, a great hypothesis. If you see about the Mendel, without him, there is no genetics now. There is no genetics at all. So he coined the term the invisible factors. Something is there which are responsible for transfer of characters from one generation to the other generation. Please remember, he is not a botanist. He is not a zoologist or he is not a life science scientist. He is by his virtue of his career, is a physics teacher, and later become math, and then fascinated with the biology, and he has shown a great world to us. Coming back to Mishra, he is a medical graduate again. Why I'm telling this all these three names is these three people has done all these discoveries in the span of 10 years, in between 1856 or 1866 to 56 to 66. Within those 10 years, they have done a marvelous experiments, and I don't know how much time Mishra might spend into the lab, because there was no great lab during those times, and they had only a lab called as tissue chemistry lab by his boss. So he was told to go and collect the bandages and take out the puzzles and do experiment. That's all. And he was so clever in identifying what solution I can add into that, so that I can take out, I can precipitate, I can see the DNA. They do not know what is DNA, but he thought some acidic substances out there because of phosphorus or rich, or say, then he made as a nucleic acids, and from there there's no need for me to tell what's went on. It was a great discovery in the history, I can say. So from there, the structure of DNA to the manipulation of DNA, we call as genetic engineering, and followed by lots of bioinformatics, lots of sequencing, lots of gene editing nowadays. I don't know where we will go in future. We say designer babies, we say precision medicine, we say lots of things in coming days. It's all happening because of this DNA discovery. Lots of mysterious things has to be explored in coming days. So friends, stay back with us and try to enjoy the session. I request everyone to stay until the complete of sessions. It's not like joining and going in between, and see that don't think that no one is watching us. Okay, we can go, but no. We are bringing senior most leaders in this DNA field. So they will share all the experiences within 1 or 2 minutes. A senior most professor, Rakesh Mishra, who we feel that is a great moral biologist in India. He is joining us maybe in 1 minute. So thank you very much. Over to Dr. Pauli.
Thank you so much, sir. Is there a resound again?
No, ma'am. No, ma'am. Go ahead.
Is it fine?
It's good. Yeah.
Yeah. Thank you so much.
Oh, now the resound is coming again.
Okay. Oh my God. Just a minute.
All right. Am I clear now?
Madam, దీనికి ఒక ఫోన్ బుక్ పిన్ ఇస్తారు మేడం, మీరు అది గుర్తుందా మేడం?
Great. Maybe I'll take over. So Praveen sir, thank you so much for telling. Really, I mean, these are the basics that people don't usually tell. I think these are the amazing things that many people have worked on. I'll just put a spotlight on myself. Many people have worked on DNA, and we have seen a lot of discoveries, and it's amazing that you shared the stories, and also the interdisciplinarity of the sciences that you shared. Like, you don't need to be a life scientist, but you can also come from other things. And also, being a life scientist or the BSc or BPC student, you don't need to restrict just being a teacher. You can also be anything, whatever you want. Cool. So thank you, sir. And now we will wait for Rakesh Mishra sir. So before Rakesh Mishra sir joins the call,
I think he joined, Jagdish. Sir
joined?
has joined. I request Shiba to give the co-host permission to Rakesh Mishra, sir.
All right.
So that, yeah. There's no resound now.
Yes, you're good, ma'am.
Yeah. So welcome, sir. Welcome, Doctor. I welcome Dr. Mishra, sir, to the event. Shiba, did you give permission, co-host permission?
Yes, ma'am. It's already given.
Yes. Okay. Great. So now may I request Dr. Purnima from the organizing team to introduce our keynote speaker, Dr. Rakesh Mishra, sir, to the participants. Over to you, Purnima, ma'am.
Okay, ma'am. Yeah. Good morning, all of you. Am I audible? Hello.
Yes, ma'am.
Yes, ma'am.
Yes. Good morning, all. It's a great honor and privilege to be part of this momentous conference of DNA 150 years. I'm Dr. Purnima from the part of organizing team. A very warm welcome to all of you. The will to win, the desire to succeed, the urge to reach your full potential. These are the keys that will unlock the door of personal excellence. I quote, it is of Confucius. The molecule which acts as a basic substance of heredity, a biological computer program over 3 billion bits long that spells out the instructions of making the basic building blocks of life. The key speaker for this session, who is a distinguished emeritus professor, former director of CSIR, CCMB Hyderabad, currently working as a director of Tata Institute of Genetics Society in Bangalore, recipient of J.C. Bose Scholarship, having completed his PhD from the University of Allahabad, expertise in the research front of genomics and epigenetics, of how information is encoded in genome during the development and its expression. An author of his famous book, Chromosome to Genome, embarked research career at IISc Bangalore. Ladies and gentlemen, we are here to introduce the one and only Professor Rakesh Mishra. Please join hands in giving a great round of applause. Thank you, sir, for generously accepting our invitation to speak at the conference. We now welcome you to take the session forward. Welcome, Professor Rakesh Mishra. A warm welcome to you.
Thank you. Thank you, madam.
Thank you, sir.
Thank you very much for inviting me for this conference. And at the outset, I'd like to apologize for messing up with the program and troubling you. I know you had to change the timings of this, and it was my mistake because I had committed to two things in the same go, and I just couldn't change it at the last minute. It was so nice of you to accommodate in the timing and shift my talk an hour later. If I can share my presentation.
So you can use the share. Yeah.
So is it now? Can you see the presentation?
Yes, sir. It's clear. We can see it.
Okay, perfect. Yeah. So we are set to go. So I appreciate Telangana Social Welfare Residential Education Institution Society, who are organizing this very timely meeting in collaboration with Dart Smart and FAVA. And the moment I was contacted, DNA is such a fascinating molecule for me. I was very excited to participate in this event. And I'm going to, so I have around 40 minutes time. I'm going to talk about a little bit about what we know about this system, this information system. And I will talk a little bit about what we are trying to understand in this complex information system that living organisms utilize. So, as you all know, and the title of this meeting shows that DNA is reaching, has reached 150 years. 150 years because it was in 1871 that a Swiss physician actually first discovered this chemical, and which eventually became the basis of science of genetics. Even at that time, although its function was not known, but Mishra suggested that it might be involved in the transmission of heredity. So he was that visionary. So halfway after this time, around 70 years, this revolution happened when the structure of DNA was solved. And this was not just simply another structure in biology. It was something which was really I would say the basis of molecular biology that we know now. The features like there are 4 nucleotides which are making a long chain, and that it is there is a complementarity in this chain, so it is a double-stranded. And because of the stacking of the bases, this is not a ladder, it is a helix that stacks, and that stacking creates many features like minor groove, major grooves, and various other things. And that is why this molecule became such a fundamental basis that it could give rise to the variety and spread of life on this planet as we see today. One of the immediate implications of the double-strand structure of the DNA was that it can copy itself exactly the same way and make from 2 copies of the double helix. So exact copying is the very fundamental, very beginning of life, and that's how the life originated, that you have something which can replicate itself, and that's the basis of life, replication, or making its own copy, or making your own progenies is the fundamental. That's how we are different from other matters. And this structure gave the very basis of how this happens. The DNA is confined into a compartment called nucleus. So that's the home of the genome. And what you are seeing here is that when this is a Drosophila cell in which nucleus has been isolated, and this nuclei has been treated with high salt so that the amount of the proteins that are involved in packaging the genome have been extracted out. Now this genome cannot be contained within the nucleus of the cell. So what you see red meshwork is actually lamine meshwork, which gives the structure to the nucleus. And what you see blue is the DNA that is oozing out of the nuclei. So it's so much of DNA that it can't be contained in a small volume. Containing the packaging was important. And the green thing that you see is also DNA, but it's a simple sequence repeat. We are visualizing it by some other means, and I will talk a little bit about the simple sequence repeat. The reason I'm showing this to you is DNA is a 4-letter language, but it's not only the linear sequence of the DNA that has all the information. Of course, that is the basis, but then that leads to many other avatars of this DNA, which is there in the nucleus. And this complex diagram just shows that within the nucleus, you have inactive part of the genome, you will have somewhere active parts of genome where things are being transcribed, then they're being processed, and this RNA goes out for making proteins and so on. So lots of complex things happen, and I'm going to give you a glimpse of how in 150 years we have learned about the DNA, how it functions, and how it gives us the beautiful life variations around, and how now we are reaching a point we can use this information for our own benefit, for agriculture or for the health. This is another thing. So for many students, it may be good to know that although this is the nucleus of a mammalian cell, oval structure that you see, and if you find a way of chromosome specific, so all of you, I assume, know that DNA makes chromosomes, which are in humans, there are 23 pairs of them. Different organisms may have different number of chromosomes, and we get one set of chromosomes, 23 of them from father and another 23 from mother. And that's how we have two sets of all the chromosomes. And these chromosomes are accommodated within the nucleus, but they are still having their own territory. Like the green thing, you see two here, another two here. These are at mitotic stage, you will see these two packed chromosomes here. So which means that unlike as it appears from here, chromosome DNA is not totally intermingled and messed up. The chromosomes have their territories and which they are properly packed and functionally well positioned for the organism to work. When we say that, it is also true that all cells in our body have identical DNA sequence. So then how it is that the blood cell behaves in one way, muscle cell behaves in another way, neurons behave in another way, how the identical DNA sequence gives these different genetic output. And one of the way this happens is that the DNA gets, so this is the sequence that you read, then it makes double helix, which gets then packaged with the help of proteins and goes and gets accommodated in the nucleus. And I showed you the picture that if you take away these histones, then this DNA oozes out of the nucleus. But each cell type must have a different kind of packaging. That's why it will have different sets of genes which are available for function, and therefore these cells are of different kind. So just to put in more simple terms, all organisms start their life as a single cell, which it can be a stem cell or a zygote. And then there are series of cell divisions or cell cycle that it goes through, and there is gradual and stepwise differentiation, and eventually you have formation of organs and organisms. And it requires a complex network of genetic elements. Imagine from one single little ball, whole bird emerges one day, or a human or an elephant emerges one day. So that all this information was there in one single cell. And which is in the genome or the DNA of the organism. So the genome is the IT hub for the making and functioning of the cell types, organs and organisms. So it's so this much we know, but how this happens is not known. And even more fascinating thing is, which I will show you in this slide, is that we know that DNA sequence has the information that makes the genetic basis of life. We know it because if we change the DNA sequence, we can change the way animal looks or behaves. It's possible. So that means the information source is there. How that information is interpreted by the system, we do not know. Diversity of the life forms are due to differences in the genetic makeup of the genome. So there are small changes, and that's why things are different in different organisms. Even within one species, let's say humans, there is genetic diversity. All our genomes are not identical. We have certain differences, and I will explain it a little bit. So here is one of my favorite drawing, which shows that life originated somewhere one time, several billion years ago, actually 3.8 billion years ago. And then it differentiated, varied, and so on. And then there is a link, branched out various, so these made plants. There's a branching here which made fungi. Here there's another branching which made the mammals and other animals, another branching for insects and other microorganisms and so on. It does two things. One is that all of us are related. We all are relatives of each other. Whether you talk about lion, you talk about a tree, you talk about a bacteria, smaller organisms like insects, we are all related. If you go long back and all, we'll find that we are distant brothers and sisters, distant cousins in a way. So that is relatedness. But at the same time, each time there is that's another beauty of life, is that each time when new organisms are formed, there is some small changes in their DNA sequence, and there's a variety. So that variety leads to speciation. So you have different species, and within the population, there's a variety. So, and those are something which are now helping us understand the biology and also how we can make our lives better. So, as I said, genome and organism's complete set of DNA. And as we now are aware that the information content in the DNA sequence is the biological, and its biological output is the basis of all forms of life on the earth and substrate for the evolutionary process. This is important. It's not only that this is how we behave and function. Any change that will be happening in the future will make the new variation or new species. And we can now, with the new technology, we can read it. We can read our genomes. That means we can sequence it very easily. And not only that, we can even edit it. We can go back and change it at our will. That has given us unprecedented power to do things which were not possible 10, 15 years ago. That's why genomics is taking the center stage, because now you can, any organism you can take and you can read the genome of this and modify it. So this is something extraordinary. So since we are so powerful with this, we must be aware of what the genome is. So for the genome era, we must understand what is there in it. One thing which I just want to impress you is that this is our planetary system. This is the sun, and here is the earth, and these are other Jupiter and other planets. Each one of you independently as an individual have enough DNA that you can go 60 round trips from earth to sun. That is the length of DNA all of you have, each one of you. That is something extraordinary, mind-boggling to even imagine. We have 3 billion base pairs in our genome, and a diploid cell, if you stretch out, it is about 3.5 meters. And this 3.5 meter material is packed in a 10 micron diameter ball, which is the nucleus. And since we have about 50 trillion cells, this adds up to 60 round trip from earth to sun. So, so clearly DNA is packed, twisted, wrapped, and folded, very compact in the cell. I just showed you earlier, and this is the same kind of picture that's packaged with the of histones and put in this thing. So the big question is how this DNA is transcribed, replicated, if the packed so tightly, how all this thing happens. In other words, how this information is accumulated, stored, maintained, and expressed in the DNA-based information system of the life. So we know a lot about DNA, but you can see how big these challenges are and how fascinating it is to solve even a small component of this, and how rewarding it will be, as you will realize as we go on. So one thing we can do is, because so much of DNA, so one way of knowing is to compare different kinds of organisms. So here you see this is the yeast, this is the worm, this is the insect, and this is the human. If you compare the genome size, you can see genome size is increasing. Very understandable, because more complex organism, you need more information, so you need more genome size, which is true. That's what is observed. But what was surprising is when more and more genomes became available, that whatever you are increasing, actually you are not increasing the coding part of the DNA or the protein coding components of the genome. In fact, it is decreasing if you look percentage. In simpler organism, much more, a higher percent of genome is coding for protein. In humans, it is less than 2%. So a number of genes are more or less same in all of them. So the DNA required for a gene is several fold more in complex organisms than in simple organisms. Complexity we mean by number of cell types, that humans have about 210 different kinds of cells, while the fruit flies have only some 50, 60 kinds of cells. So they are less complex. So when evolution has taken place from simple to complex organism, what does it tell us? What is the outcome of evolutionary process? The outcome is that static number of genes number don't change so much, but there's more non-coding DNA, that is more DNA per gene, which means there is a parallel to increase in the complexity. There's more DNA, more if complexity is more. So what is then the function of this excess DNA that is being added up? We are made of proteins. If they are the same, then what is the more and more DNA doing? So So reading the information content, that means we have to go beyond what is the protein coding, because that tells only 1.5% of our genome. So one conclusion we can make safely is that evolution of complexity is not by more genes, but is by more sophisticated regulation of genes. It's like you have cement, steel, bricks, and sand. You can make an ugly hut, or you can make a beautiful multi-story building if you are a good engineer. So it depends on how good sophisticated your engineering is. The same way, same amount of information, material, which is the protein coding genes, can be used to make sophisticated organisms if you have more sophisticated regulation. So what is evolving is the regulation, not new building blocks in the life. And other thing is that this complexity, another way of looking at it is that each cell type has its epigenome, what we call. So genome is identical, but epigenome is different. That means same genome, but performing differently, packaged differently, modified differently. And then there's a, which means there are multiple ways of packaging the same genome. So like a human genome, which is identical in each of our cells, can be packed in 210 different ways, so that you have 2 different 10 cell types. So the non-coding part of the genome, which means the genome that does not code for proteins, contains these regulatory elements and has the cell type a specific packaging code, and provides grammar to the genomic language, if you want to say in another word, and uses the subnuclear compartmentalization and interaction with the environment. All those features are built in this excess DNA that is being accumulated during evolution. So similarly, the functional version of the genome requires that, as we just discussed, genomic packaging. So differential accessibility of genetic loci, like for instance, in muscle cells, you don't want genes that are involved in thinking to be transcribed. Similarly, in brain cells, you don't want proteases that are used for digestion to be made. They need to be made only in intestine cells and so on. So there is a packaging code which allows different accessibility, and that defines cell type. Then you need packing material for this. How you package it? DNA by itself cannot be packed. You need histones and non-histone chromatin proteins, and they're a very fascinating discipline of biology. And they are remodelers and non-coding RNAs that are involved in this process. Those are emerging fields. And in addition to the third component is the nuclear architecture in which this whole genome is packaged, how it is anchored to a different parts of the nucleus, how territories are defined within the nucleus. And many of these questions we address in different ways in our lab. And one example which I'm going to give to you is about in our quest to understand the function in the non-coding part of the genome, is to ask what do simple sequence repeats do? Do they have any function? So simple sequence repeats make about 3% of the human genome. Remember, protein coding is only 1.5%. This means this is twice as much in terms of quantity of DNA. So what do they do? So one thing we did in the lab was now advantage we are having is lots of genomes are being sequenced and information is publicly available. So we are now looking, let's say this is a tetramer. So it's a 4 nucleotide, and there will be, these are all possible tetramers that we can think of. And these are different organisms. These are simple organism. I mean, this is a human and this is a simple organism, the other side. And this is the abundance of the particular repeat. So for instance, you see that this here is AG80. This is present in all of them more or less. In some of them larger quantity, some of them small quantity, but you see this tower in the entire row. But many of them, this one or this one or this one or this one, let's say CCGG, is not found anywhere except in some simple organism in small quantity. So that means among the all possible simple sequence repeats, in this case here is tetramer. This is true from all the simple sequence repeats. Only few are chosen, and once they are chosen, they are there in all the organisms. Those who are not chosen, they are absent everywhere, right? So that means there is some selection operating. The other thing what we notice is that if you take each one, let's say AGAT you take, so what you notice is that here this is the size of the repeat, this is the occurrence, and different colors show different organisms. So what you see is that this is the size which is preferred in most organisms. We will not discuss this one, which is zebrafish. So that means smaller than this is not liked by the organisms, and larger than this is also not liked, and therefore you have a preference of certain size. So that means there is a preference for the size and preference for the sequence. So that means there is some selection pressure operating, and they are useful. That's why they have been enriched, and not all the sequences or all the size are there. There's no randomness here. So this was our major finding, that SSRs have unique selection pressure operating. But the challenge was, what is it, the selection pressure? What are they doing? We know they are not protein coding, so they are not coding any proteins, so they're not doing anything. They must be involved in some kind of location, some packaging, or some regulation. And I will not go into many details, so what was the basis for thinking this way, but we made a hypothesis that these repeats, since they occur in many places, they may come together, and that will allow the DNA to loop and make different domains or functional compartments, where an active compartment is one side of the loop, other compartment is the other side of the loop, and you have in here the packaging done by these repeat sequences and the proteins that interact with them. So our first prediction was that then these repeats should function as regulators. And we did this in an experiment to show. This is just for the students to realize that if you have idea, you can, lots of technology is now available to address your question. So this experiment is done in fruit fly as a model system. So we take a fly to begin with, its eyes are white, and we introduce a gene in this fly, which is this red color shown here. So eye becomes red. And this is the embryo, which normally will have no blue color. But because we insert this gene where this regulatory sequence drives the expression of the blue gene, so you see blue stripes being formed. So that is the system. Now we take a repeat, AGAT, as I just mentioned. It is the same construct is there. Now what we have done is we have inserted between this oval and this blue bar a repeat. This repeat is taken from human genome and put in the Drosophila system. Now you see that red color is fine, the eye is red, but the embryo now does not show stripe or shows very little stripe. So now imagine these are transgenic flies, and it's an in vivo system, it's not an in vitro system. Now you use genetic trick and you just pinch off this AGAT repeat from here, and it becomes more or less like how we began there. Then what happens to the animal? It is red eye as everywhere. The blue color comes back, which means now this oval again starts to talk. So that means in this fly, there was nothing wrong. There was no mutation, no other problem. It's just that you inserted in between something which prevented the crosstalk between these two elements of the genome. So that means these are the repeats are useful in packaging the genome and creating independent domains of activity, so that a gene, let's say here is a muscle gene and here is the brain gene, they don't have to interfere with each other. They can live in their own autonomy. And so this was that's why you have so many simple sequence repeats, because they are required for this. So in fact, we did the most massive analysis of simple sequence repeats, and we generated so much data that it started to look like an art of life. So we call it simple sequence repeats, art of life. And this is the data. This is very very pleasing to see how you can interpret your results. So I will just explain. It will take 1 minute to explain. So here on the x-axis, you have each column is a repeat. There are 501 different kinds of, there are 501 different columns. And we have used here 37,000 genomes, which are on the y-axis. And we have arranged them like these are land plants, these are green algae, ascomyces, so on, mammals, and so on. So these are there, each one, and 501 repeats. and color code when red means absent and black means present or higher quantity, higher abundance. And so that is the data. So, and you have 4 billion SSRs total together. So here you are seeing 4 billion dots organized in one side, in one axis as a repeat and other axis as an organism. So what do you see here? It's not a painting, it's a science. So what you see here, you see pattern that some repeats, like these are the, of the 500 ones, some kinds of repeats are liked by all the organisms, most of them, except a few. So some repeats are preferred, which I showed you in the beginning in the graph, that some repeats are preferred and others are not. So these are the repeats which are preferred, and these are repeats which are less preferred. Then there are organisms which, like, for example, this Ascomyces, this group doesn't like repeat at all. So there you see all the time, most of the time it is red and then something green enough. So that means there is no randomness. There is a very clear pattern, like this organism in the green algae, it doesn't like repeat at all the way you see red line. Then what we are now finding in detail analysis of this, sometimes you see here a black dot. So this means this is some mammal, and this is a particular repeat here. This is preferred by this organism, is there. It's not there in any other thing. And there are many such black dots you can see here and some other places. So this, what is the conclusion from this analysis? This shows that these 3% of the genome what makes simple sequence repeats. There is taxon-specific variation in exonic, intronic, and intergenic densities. Is a highly constrained in organisms with heterogeneous cell types and greater diversity in motif abundance, density, and GC content in simpler organisms. So if the organism is simpler, there is a lot of variation. But in the complex organisms, their length is increased and variation is reduced. So that means these sequences have got constrained in developed organisms. So based on these observations, our proposition is that simple sequence repeats are integral components in speciation and evolution of organismal complexity, as they bring coordinated genome regulatory features, which I showed you some data about that. And this is something new, and now more and more sequences are coming. So we are doing lots and lots of such analysis. Now I will switch gear a little bit and go to large scale data generation or population scale genome and what are the uses of this. And in India, there are lots of programs which are generating large scale data of human and other organisms. Just to show, to convince you why it is important, is that if why population scale human genome should be, if all are the same, we have same genome, why to worry about it? So we have 3 billion, there are 3 and then 9 zeros. These are the variation point. These are the nucleotides or bases in our genome. 99.9% we are same, we are identical to each other. But 0.1% is variation. This 0.1% means 30 lakh variations are possible between any one of us. That is what makes, though we are so similar, we have genetically distinguishable, and that's essentially what we call as genetic diversity, and that gives lots and lots of opening to understand biology and many other utilities. Some are indicated here. So implications of genetic diversity in healthcare, for example, is very useful for diagnostics, is used for prognosis, which drug to use, which treatment to use, and pharmacogenomics, which I will explain a little bit toward the end, that one size fits all is not applicable here. We are all unique, and we need unique treatments of our disease and other conditions. So now, since we have the genome, the same DNA which were discovered 150 years ago is now giving insight into our health and agriculture and many other interventions that are possible. It's now becoming the basis of precision and personalized medicine, rare genetic disease, and so on. India is very unique in this context, from the genome context, and we have our specific problems. Uniqueness of Indian population is that we have 4,600 ethnic groups. These are mostly endogamous populations, and therefore there's an accumulation of ethnic variations in this, because we have a social system of marrying in certain groups and all are not marrying outside. So our genetic diversity is there, but it is confined to uniquely to different groups. So because of that, population-specific mutations have become what we call as founder mutations. They have been accumulating, and therefore there are population-specific diseases and drug response. And this is something which is very, very important. Here I will give you one example to really impress you the scale of this problem. So endogamy practice in India, we can discuss at some time, started 2000 years ago. And that has led to population-specific diseases. Today, we have unsustainable burden of rare genetic diseases in the country. Many times we don't notice it, we don't talk it, but this is the fact. So here are the numbers. Any given time, number of Indians suffering from rare genetic diseases are 70 to 90 million. Okay? That's the number. And for these people, we don't do much. Because treatment costs, even if 1% of these people are to be covered, the treatment cost for them is 1 lakh crore per year. 1% of this, if you have to treat 1 year, it will cost that kind of money. So it's obvious that there's no question that we are going to manage. And in addition to that, of course, there is no real final specific treatment. And there is a lot of family stress, social trauma, productivity loss, and so on. This is a huge scale, okay? And several hundred kinds of such diseases are yet to be discovered. As I said, because of our unique ethnic groups, we have this problem. But there is solution also. So much of this can simply be avoided by targeted genetic testing, prenatal diagnosis, and genetic counseling. So we can just prevent the birth of, or reduce significantly the birth of such patients or babies, and we can solve the problem. There are other ways of doing it for which, but their cost becomes prohibitive, the gene therapy and so on, and they are not even accepted at the moment. So this is the scale of the problem. There's other thing which is very, very common, but we don't attend at the moment, but doctors are familiar with this, is what we call as a personalized medicine. Here are examples. Is the percent of patients for whom the drugs are ineffective, right? So for a depression, 38% of the people don't respond to the A drug given to them. 40% for asthma. So many things. Go to cancer, 75% of the people will not respond to A given drug. 25% will respond. So which means, but that doesn't mean that these who are not responding to a particular drug will not respond to anything. Some other drug they will respond to, but it's just that we do not know which one they will respond to. So this is called personalized medicine. Once we know their genomes, then we can figure that out. So this is what is explained here. So each individual is genetically unique because our DNA that we have is unique. You remember 30 lakh point of variation. So therefore we have our genome is like our Aadhaar card of life. So when we are in a crowd, we are saying two kinds of people, let's say. And traditionally we use one size fits all. So same patients are given same diagnosis and they receive the same treatment. But if we use the genome privileges, then based on genome sequencing and genotyping, we can classify them into, let's say, genetic profile A, genetic profile B. Now we know that A will respond to medicine 1 and will not respond to medicine 2, and B will respond to medicine 2 and not respond to medicine 1. Now we don't need to treat them separate the same way. They will get different treatment, targeted therapy. They will get different treatment, let's say standard therapy and so on. So now you can imagine a cancer patient who goes through chemotherapy, does not get the benefit, but goes through the torture of side effects and all that. That can be totally avoided and it's very expensive. So if we knew and apply this genome-based personalized medicine approach, we can not only save money, but also make people more comfortable and have better treatment. So many times, years ago, Hippocrates said that it's far more important to know what person the disease has than what disease the person has. So depending upon who you are, your treatment is going to be different. That's the era we are entering into. That's where the DNA sequence or our genome information is going to be applied. What are the technologies that are required for the future? Although I said something, but it's not, you need a lot of information for that. For example, all of us have to get our genome sequence. At the moment, it's expensive. Right now, it can cost, depending upon how you want to do it, sometime maybe 70, 80,000, maybe sometime 50,000, something like that. Will it become cheaper? Will it become 5,000? Answer is yes, very likely. So those are the few of technology I'm giving. These are not the dreams. These are technology we are there. They are already there, and they will become applicable at large scale in future. You are going to have handheld battery operated sequencers. So you don't have to bring your sample in a huge sophisticated facility. You can do it with your laptop and mobile phone, attach the little battery operated machine, which you can keep in your pocket and do a sequencing there. Since that machine is connected to your mobile phone, it will generate and transmit the information through the cloud sharing internet to a server from which a doctor will be, a clinician will be able to see that. And your computer now knows so much of genome and so on that it will analyze and delivery of the result to the user. So in the village, you can be doing this, and next day you can get the information what treatment should be given to you. So that is the era we are going to enter. Affordability, as I said, will not be an issue because it will become cheaper and cheaper. And in fact, you may get even paid if you for the consent, that if you allow your DNA sequence information to be used for research or for some other data mining or modeling, you can even get paid. You go and get your DNA and get some money and you can come home. We need what lacks at the moment is we need policy, we need regulatory setup and accountability that need to be resolved. Because even if it is anonymous, if we know that in this part of Andhra Pradesh or this part of Telangana, people are more susceptible to a particular disease, whether you know it is individual or not. But then insurance companies will not like to insure those people because they know that they will have to pay for their hospital bill. They are more susceptible and so on. So, and if this information is available at individual scale, sometimes companies may not give a job to someone because the person is more susceptible. May get disease or not is a secondary thing, because many things are multi-loci and environment plays a role. So we need to have robust policies, regulatory system, acceptability and accountability so that we can use these kinds of potentially extremely powerful information in our daily life of taking care of medical needs. We need more technologies. As I told you that we can sequence in very fast speed. Within a few days, you can have your whole genome sequenced and analyzed and ready for use. But we need more genome, transcriptome, proteome analysis possible at single cell level. As I told you, the muscle cell is different than brain cell. So the transcripts that are made or proteins that are made or other omics analysis, if it can be done single cell level, that will be more effective. And we need high-end automated analysis and data management, mobility, safety, because those things are manually very, very difficult to do. And then we need genome editing of very high accuracy. We can do genome editing, but there are side effects of that. Somewhere if the wrong site is hit, you may get different kinds of result, but we are more or less, this should be shifted in this box, because this is already there in the future. Is not something technology is needed, it's already there. So this is my last slide. So what is coming? And young people can dream be part of this change which is coming in, let's say, in the field of agriculture. That will now take the advantage of genome technologies. You will have designers, crop, plants, and farm animals. So, for example, your crop will require less water, resource usage can be controlled. Now we grow rice in a field filled with water. Soon you will have rice that will grow without filling water. It will grow like wheat or any other millet or other crops. Less environmental impact. So you will have crops which will not need insecticide. Insects will not eat those crops. We can bring those resistance in the plant. We will have plants which will provide higher nutrition. They will have high protein, low glycemic index, more iron, zinc, which is required. So the same rice that you eat today will be make more nutritious and low GI, which means even diabetic people can eat freely. So this is about the agriculture. You will have pharmacogenomics based prescriptions. It depends on your genotype or your genome signatures. You will get what kind of medicine, what time of the day you should eat the medicine. This will be told to you along with, and how much of the medicine you need. I may need 50 mg, but somebody else may need 10 mg because my metabolism is faster for that drug and somebody else's metabolism is slower. So that if they take a higher dose, it will be toxic for them. But if I take lower dose, I will have no effect because it will get eliminated from the system. Similarly, you can have, if let's say your medicine targets a protein in the body, a receptor that is expressed in the night. If you take in the morning, the whole day, your drug is of no use because that protein is expressed only in the night in the body. So that you should take in the night, not during the day. So, and this is known that more than 50-60% of the drug targets have circadian rhythm in their expression. So these are the sophisticated biology that we are understanding, which will make the future healthcare very different. Rare genetic diseases and other genetic diseases we will be able to prevent and eliminate, as I described how to do that. Cancer and psychiatric drugs, they will need personalized and precision medicine. They will not be given hit and trial and 75% not responding, 25% responding. And something else which I did not talk about at all is what is called microbiome. In our body, we have a huge number of bacteria, trillions of bacteria. In fact, we have more bacterial cells than our own body, our own cells. So, and they influence our health, disease response, and so on. People have started, because of genome sequencing technologies, they have started to understand this complexity and this change during disease conditions and so on. So that is going to be an important player in our healthcare system. So as you can see from basic discovery of DNA from a pus cells of the neutrophils 150 years ago, we have reached today where we understand enough about the DNA, complete set of DNA, which is genome in our in different organisms and in humans, that we can have a better, sophisticated and efficient healthcare system, better agriculture or farm animal system where environment is less stressed, and have a much better, comfortable situation and understand more the biology and the animal and plants around us. So thank you very much for your attention, and I finished exactly 45 minutes, and I will stop sharing here, and will be happy to address any questions if there are.
Thank you so much, sir. Thank you so much. It was really a wonderful session. I really love the way that you have started from the basics of DNA, right from Frederick Mishra's discovery, and then you have talked about the current trends that are happening in the field of nucleic acid research. And what I really like is the last slide where you have given some interesting areas or to explore for the young students who have joined the session today. Really, thank you so much for the lecture, sir. I'm afraid we don't have much time for the question and answers, but I have a small point to make here from the lecture that you just delivered. So we have many school teachers who have joined the session today. And we all, I mean, as a science student, I was taught about junk DNA in the school, but after listening to your lecture, definitely I advise the teachers not to call it junk DNA anymore, because clearly it is doing a lot more than the not coding DNA. It has a huge role to play in evolution, in disease, whatnot. We already heard from Dr. Rakesh Mishra. So please refer to it as non-coding DNA, and please enlighten your students about the mysteries it holds within itself and encourage them to pursue it and to study it, to learn more about it. And sir, I also take this platform to mention about the Milo CCMB project that we are doing in collaboration with CCMB, where the students are learning more and more about the research that is being carried out there. So please encourage the students to interact with the scientists from CCMB and keep loving science and pursuing science. Thank you so much, sir, for joining us. If you have any special message to the students, just a brief one who have joined, because we have school students also in the session today.
Sure. I mean, you summarize for the school children. I have to just say that if India wants to be a strong country, if you want to be developed and respected country in the world, what we need is a strong science and technology. If we keep importing medicines, keep importing technology, keep importing tools from other countries, we are actually not independent. And many times our problems are unique, and problems can be solved only by ourselves. And during this pandemic, we have seen that how during the beginning of the pandemic, there were no diagnostics. It was costing 3,000, 4,000. Today we have diagnostics indigenous completely. It costs 100 rupees. So that means you are being looted in the beginning. And at the time of emergency, you will not even get it. Forget about it. If any country which is exporting, if they need it, they will not give you. So we have to build our capacity, be independent in these things, be indigenous and self-sufficient. And as I said, where India will stand in next decade depends upon how strong is our science base or how strong is our technology base. The way we have become powerful, power in the IT technology, the same way we have to be powerful in our other technologies like defense technology or healthcare technology or other devices technology. And this will happen only when young people will take science as a career and build this nation. Otherwise, we will make money, work for the rest of the world and import the things till somebody is willing to do business and earn a profit from us. But during the emergencies of war or pandemic or bioterrorism, we'll not be able to support ourselves. So to be strong, we have to, as a system, support science. As you have seen, what potential we have. Our genetic diseases will not be treated by others. These 70, 80 million people, America will not find medicine for them. You have to find it your own way. And similarly, in our population, who are the 75% who are not going to respond to a particular chemotherapy? We have to find out. It's not going to be tested and tried in the US. So this is our problem. We have to handle. Technology is there, science is there, and we have such a brilliant, huge number of young people. We should be really the superpower. India is one of the youngest country, and we are having all the technologies. We have demonstrated momentum. If we can send our machines in the Mars, we can do anything. It's up to people to decide. So I encourage young people to consider science as a career, because then you can make changes that our society requires. And I I'm very happy that you all are organizing this activity which motivates people, and teachers play important role because they can motivate hundreds of children every year. So you hold the key. So thank you again for giving me this opportunity. I'm very happy to be part of this.
Thank you. Thank you so much, sir. It was really a pleasure and an honor to have you with us today. Now, we also have the next speaker, Dr. Gautham Das, who has joined us. I apologize for the little delay over the program. And so may I now request Sheba to introduce the speaker to the audience?
Yes. Thank you, ma'am, for allowing me to introduce our next speaker. Our next speaker is Dr. Gautham Das. He is the co-founder and managing partner of an advanced science and deep tech startup company, Microbiome Therapeutics LLP, specializing in genomics for healthcare, pharmaceuticals, agriculture, and biotechnology domains. He completed his PhD in molecular microbiology from the Indian Institute of Science, Bangalore, and postdoctoral research in genomics and cancer at the University of Massachusetts Medical School in the United States. He has over 20 years of experience in research, high-impact international publications in Cell Press, Nature Publishing Group, Oxford University Press, PNAS, etc. Apart from the publication, he has 8 grand patents from the USA, Israel, and Australia. He has held the position of the Assistant Vice President and the head of biomaterials position at the R&D division of the Reliance Industries Limited before founding a genomics startup company in Mumbai. Sir, we appreciate you sparing your time to be with us here today, and I request you to lead the session.
Thank you. Thank you very much to the organizers. Thanks especially to Dr. Jagadish, who first connected with me and invited me to this very interesting session, what I have seen in the current session with Dr. Rakesh Mishra. The whole motivation of teachers and students, it's really important for our country to move ahead. And I agree with all the points that were mentioned. And I urge students right in the beginning of this talk to take up science as a career. And you will see at the end of the talk how science can be actually rewarding in terms of doing basic science to actually translating the science. And I'll be talking more on the aspects of translating the science for the immediate needs of India. So I have titled the talk, Genomics for All, Platforms, Products, and Services that our startup does, and in general, to resonate with the theme of DNA at 150, the celebrations that we're doing of 150 years of DNA discovery. So yeah, so before we go on to the talk, it is very important for us to understand what is genomics. We have heard the last speaker talking a lot about genomics. So I wouldn't go into the details again and make it redundant, but a brief introduction of what is genomics. You have cells, you have chromosomes, you have DNA inside the chromosomes. If it is a eukaryote, if it is a prokaryotic cell or a simpler cell like bacteria, you do not have chromosomes. And that is how the DNA structure looks like, as in this slide. And the base pairs of the DNA, all you have heard in the last talk, so I wouldn't go into the details. The study of this DNA or the study of these genes in a holistic way is what we call genomics. Now, to understand genomics, it is very important to understand what is DNA reading or DNA sequencing. To read the DNA and apply it to any setting is the whole study of genomics. The first part in all this is DNA sequencing. How do you sequence DNA? You have a tissue, organ, cell, anything like the liver, for example, in this slide. You extract the DNA, and using a variety of steps, what we call as library preparation in next generation sequencing and genomics applications, what we do is we make libraries of DNA, wherein hundreds of millions of genomic DNA fragments are placed on a surface, and then a single fragment that makes thousands of copies of DNA are inserted into a machine, specialized machine called next generation sequencers, wherein the entire sequence of the DNA that you give as an input is sequenced, and that is how the DNA is read today. I will come to how it was being read earlier, and this is how the whole concept of DNA reading. And here, for the benefit of students, teachers, computation analysis. I mean, while we were doing biology, at least many years ago, it was not thought that computation is going to be an integral part. And today, as we see, computation analysis and computation in particular is an integral part of all biology applications. Genomics in particular has to have computation analysis of data embedded within the biology. So how did the sequencing technology evolve? We had first generation of sequencers, as I just now mentioned. Sanger sequencing, if you all have heard this name, this is from the discoverer of sequencing. Sanger sequencing, Maxim Gilbert sequencing, Sanger chain termination in the old days, wherein we used to infer nucleotide identity using DNTPs, then visualize this using a series of experiments of electrophoresis using DNA fragments of 500 to 1000 base pair fragments. So that's the first generation of sequencers. And then we moved on to second generation of sequencers, what we also call as next generation sequencing, wherein the technology is a little different. Of course, we are reading DNA, but the technology is a little different, wherein it is high throughput in the sense that it parallelizes all sequencing reactions in one surface, and only 50 to 500 base pair fragments are read at one time. And this is how the companies like Solexa, 454, Ion Torrent, and the leader of all sequencing company called Illumina, as you students and teachers, you would be hearing about this company a lot today and in the future. Illumina occupies about 80 to 90% of all sequencing that goes on in this globe. And this is the next generation sequencing, or the second generation sequencing. Then comes the third generation of sequencing, which is already going on now, which is the long read sequencing. And companies like PacBio and Oxford Nanopore have made platforms which reads about 10 to 1000 kilobases of fragments on an average, and this sequences the native DNA. So the concept or the technology is a little different from the next generation sequencing that we heard about Illumina. Here, native DNA is read in real time in single molecule resolution. So these are the sequencing technology, how it has evolved from 1977, wherein we saw the Sanger sequencing, to today, wherein we are going to third generation of sequencers reading large molecules of DNA in a single molecule resolution. So what is important for any technology to be adopted more and more is the cost. It has to be affordable. And fortunately for all of us as scientists, we are really fortunate that the cost of DNA sequencing from 1977 to now has defied Moore's law first, and it has defied all laws of economics. I mean, today, wherein we saw whole genome sequence of human at $1 billion. The Human Genome Project costed $1 billion in 2003. And today you can sequence, as Dr. Rakesh Mishra mentioned, a human genome for less than 50,000 rupees. So that's less than $1,000. So from $1 billion, we are talking about $1,000 genome. That is the reduction in cost in just less than 20 years. And the technology where it will go, I am very confident that we will see DNA sequencing to be available free, as Dr. Mishra rightly mentioned, free for all very, very soon. So that's the technology, and we should tap the potential of this technology very, very soon, because I did not see such a parallel of cost reduction in any other technology. And the biggest beneficiary of all the sequencing has been the human genome. So the applications have been in developmental biology as basic science, precision medicine or personalized medicine as we call it, diagnostics, gene therapy, and the computation as I spoke. Combining all this, human genome has been the biggest beneficiary of genomics, wherein decoding of the human genome took place in less than 20 years ago. And today we are talking about treatments which are personalized and precise to every individual. So that is the advances we have made in the last 2 decades. So all of here, the main audience is teachers and students. I would like all of you to look at this slide here. This slide talks about NGS applications, or next generation sequencing applications. And I am very confident that this slide has covered everything that NGS can be applied to. And I urge you to write to me if you don't see any application here. Genomics can be applied to biopharmaceuticals, to transcriptome, oncology, novel pathogen discovery, to everything. That is the power of this technology, that it can be applied to such diverse and varied kind of fields, and its applications ranges from precision agriculture to precision medicine. So we all have to tap into the potential of this technology very, very soon. So from organs, tissues, what we heard just now in the last talk, today I'll be talking about a new technology, which is single cell sequencing. What is single cell sequencing? It examines the sequence information from individual cells, not tissues, not organs, but individual cells. And it reveals the cell population differences between two cells. If there are two cells in a given tissue, it can tell you the difference between the two cells from the sequence information. And that is very important for understanding of the function of individual cell in the context of the microenvironment, especially in diseases like cancer, etc. So that is single cell sequencing. So from where we have come from 1977 Sanger sequencing, we are now today, as we are talking, we are sequencing single cells in a population of cells. And why do we do it? In development, as you know, we have different kinds of cells. So single cell, the whole genomics and transcriptomics play a big role in understanding in detail the whole development of an organism. Also to study the mosaicism in somatic tissues or cancerous cells that take place, and in microbial systems wherein a population may not be the same as a single cell. So its applications ranges from genomics, epigenomics, transcriptomics, stem cell differentiation, immunology, nerve cell differentiation, circulating tumor cells, etc. How do we do it? There's a specialized technology, a company called 10x Genomics, wherein 10x barcoded gel beads are available, wherein emulsions are made and a single cell, sorted out cell, the whole genome sequence of that DNA is read by sequencers like Illumina. For students, it's very important to not just understand the science, but also the business part, where this whole, I mean, some of you could be interested in industry later, could be interested in becoming entrepreneurs, becoming industrialists later. So all of you, genomics, the market, how it is going is very, very important, not just the science. In 2018 to 2026, the analysts, whatever have analyzed, is that from 18 billion, we are talking about this technology or this genomics market to go to 83 billion by 2027. And this is a little older forecast, I would say. Pandemic has changed our view of how genomics markets will evolve in the near, very upcoming future. That is the potential of this technology, both in terms of science and economics. What are the drivers for this? Rising government support. Governments all across the world, including India, have been very, very supportive of genomics right from the beginning. Reducing cost of sequencing, as I said, we are very fortunate to see this revolutionary reduction of sequencing service costs, which that means that we have more freedom to do more and more experiments using genomics. Personalized medicine, yes, the promises were much higher, but then we have not got where we thought we will get there. But in future, we will soon get to personalized medicine in developed countries as well as India. So coming to our startup, Microbiome Therapeutics, and I'll take you through our journey of how this genomic startup we have built in Mumbai, India. We are a fully equipped science and deep tech startup, as introduced in my introduction. Thank you. We specialize in genome sequencing, diagnostics, bioinformatics research, and services. We are backed by a big industrial group in Mumbai called Excel Industries, who have experience in industry of more than 75 years. We set up a laboratory in 2020 in the middle of pandemic with state-of-the-art genomics and diagnostics infrastructures like Illumina, Oxford Nanopore, platforms, next generation sequencing platforms, real-time PCR, advanced computation, and bioinformatics. And I'm very happy and share with you all that in a very short span of time, our company has generated over 1 trillion bases of genomic DNA here, doing services and research on our products. We have processed over 750 genome samples and delivered over 35 research projects to government clients, private clients all across the country and abroad. We have a team of, we are a small team, we are a startup. We have a team of about 10 plus, to be precise, 13 experts in field of molecular biology, computation biology, next-gen sequencing, microbiology, and biosoftware. As I said, software is an integral part of our company and any genomics to be. So this is the team. we have Ravi Shroff, who is my co-founder, and Dr. Nandini Dasgupta here, who leads our science. And we have a team of scientists, business professionals, technologists, etc., who combine the compose the whole team of my biome therapeutic startup. And interestingly, I was asked by the organizers to talk a little bit about my own journey of a startup, which young students from school and college might find interesting. So I did my bachelor's and master's from Hyderabad, University of Hyderabad and Osmania University. Then I went to PhD in Indian Institute of Science, Bangalore, wherein I did molecular microbiology. And then I did postdoc at University of Massachusetts Medical School in the US, doing cancer biology, genomics. And that is where my whole interest in genomics started. And then I joined Reliance Industries to lead their synthetic biology department and genomics department. And there we were doing very interesting and applied genomics for biofuels, renewable energy, technologies for the future. Reliance has invested in technologies for the future like renewable energy, etc. And 7 years into corporate, I moved on after that to start my own startup along with my co-founder. So we started MyBiome Therapeutics around 2020 in the middle of pandemic. And we are a little over 1 year now doing genomic services and research for varied research services, products, platforms. So this is in short my journey from science, basic science to translational science, and now actually building a company, genomics company here in India, having a very high-end lab in Mumbai. What do we do in our company? We focus a lot on problems specific to India. As Dr. Rakesh Mishra also mentioned, we have a unique set of problems. So infectious disease is a big problem, is a big burden in India. So we focus a lot on infection control and genomics for infectious diseases, for bacterial, fungal, COVID, we have done a lot of work, Mycobacterium tuberculosis, extreme drug resistance, TB, etc. Hospital microbes, which cause hospital-acquired infections. We focus a lot on the wellness also, wherein we have a product on gut microbiome and many more products on skin microbiome, oral microbiome, vaginal microbiome, respiratory microbiome we have developed. We focus on India-specific panels for cancer, reproductive science, inherited diseases, rare diseases, and then immunology specific to India, Indian genome specific information. So that is in broad our core areas of research in our startup. So what have we done in the past 1 year in academic front? We have been very active and we have already published a peer-reviewed paper in Frontiers in microbiology, 2 book chapters wherein, as you can see on microbiome in Springer Nature and Elsevier. We have applied for 2 intellectual property patents in India. And then we have contributed to many preprints in the COVID arena. So to start with, something that we do in the company is NGS, next-gen sequencing for infectious disease control, as India is a big burden of infectious diseases. I wouldn't go into details of this. You can have airborne transmission of pathogens, contact transmission through water, transmission through vectors like malaria and dengue, etc. So overall, infectious disease is a big burden in India, and all of us have seen the COVID pandemic and the interest and the importance of genomics in infectious disease control. How this is done in earlier days or even today, culture and microscopy becomes the gold standard of all infectious disease diagnostics. But it is time consuming, it cannot detect non-culturable species, and it is always dependent on a priori knowledge, and there are errors. So now comes polymerase chain reaction, and all of you must have heard about real-time PCR in the COVID pandemic. It has greater speed, lower cost, higher specificity, sensitivity. It depends on predefined targets. It cannot detect novel organisms, and it cannot differentiate between genotypes. On the other hand, next generation sequencing, or NGS as we call it, is high speed, scalable, high specificity, sensitivity. It can detect anything, any non-culturable species, novel organisms, genotype differentiation, but it is higher in cost, but fortunately the costs are reducing drastically. This is going to be the future. Antibiotic resistance can be measured, can be predicted using next generation sequencing, and we do this in our company. How antibiotic resistance occurs, if college students, teachers know, that you have a small population of antibiotic resistant organisms and that multiply to cause a whole population of antibiotic resistance. In India, it is especially a bigger problem as we see over-the-counter sale of antibiotics. We have more communicable diseases than the West. We have a poor healthcare system, which is improving thanks to the government spending. And so this problem is more prevalent in India. We have to use specialized tools like NGS in antibiotic resistant strains. And this is the same slide wherein we saw culture and microscopy earlier for antibiotic resistance. PCR came next. And now we are using NGS for antibiotic resistance mapping or predictions. Tuberculosis in India is a big threat. 10 million people fell ill with tuberculosis, out of which 1.5 million people die every year. And drug-resistant tuberculosis is a bigger problem, wherein, as you see on the left, you have rifampicin, isoniazid, etc. And if none of these drugs work, what would a clinician do? Extensively drug-resistant tuberculosis, or XDR-TB, is a form of tuberculosis caused by bacteria that are resistant to some or most anti-TB drugs. Now, this is a big problem. And India is a big burden of drug-resistant tuberculosis. So we are addressing this problem using NGS, wherein we do rapid drug-resistant testing instead of growth-based drug susceptibility testing as it is done phenotypically using culture. That takes very long time, around 4 to 6 weeks it takes if you do a growth-based drug susceptibility testing. NGS methods are very accurate, and it takes only 1 week to actually tell the drug resistance of about 22 drugs that we are doing in our company. So it is a cultureless method, it is unbiased, and then it can take any fluid like bronchoalveolar lavage, cerebrospinal fluid, ascitic fluid, biopsy, or any clinical sample. What is interesting is novel India-specific mutations can be queried as we do more and more research into TB. Novel mutations we have been able to find. And we have also developed amplicon-based sequencing wherein it is cost-effective because it only does a small amount of sequencing, and through which multi-drug resistance, XDR, TB, we are able to detect in our company. Early detection of TB is very important as it guides the clinicians in the appropriate adjustment or personalized preventive medicine, as you call it. This is how the multi-drug resistance NGS profile looks like. So you have all these drugs, and the doctor can choose, like if it is resistant, we color code it red. So this is not to be used in the clinical practice. So this is, we have been seeing very good response from the clinical community, the hospital community, and the diagnostic community that NGS can be more precise, accurate, and faster than the growth-based assays that take 4 to 6 weeks, which is a huge time in a TB patient's life. What did we do in the COVID-19 pandemic? All of you saw that for the first time, genome sequencing has helped guide the public health response to a pandemic in near real time. In diagnosis, vaccine target selection, genotyping variant, drug target selection, everywhere genomics and whole genome sequencing was applied. And this is where epidemiological surveillance for the first time has seen the application of genomics, the whole variant calling, variants, etc. I wouldn't go into the details of this, but we have made a solution in our company where we don't do whole genome sequencing because it is expensive, and to use it for a country like India is going to be very, very expensive. So we made a very small tool, a small amplicon sequencing tool, which detects all the variants of international concern, which is only 14% of the whole genome, and therefore it is cheaper. And the required predominant information, over 75% of all these variants that we hear in the news is covered here. So this is a snapshot of how it looks like, and all the variants that we hear are being covered in our sequencing panel. So this is how it looks like. You have all the lineages, and our panel covers all these mutations that we hear routinely in the news. Next, I'll shift gears to another research area that we have been doing in our company called Gut Microbiome. And gut microbiome is the totality of microorganisms, bacteria, virus, protozoa, fungi present in the gastrointestinal tract. It identifies all the microbes using a stool sample. And what is important is the application, that changing lifestyle and diet improves gut microbiome. We cannot change our genome, but we can change our microbiome by proper lifestyle and diet. Addition of prebiotics and probiotics in our food and lifestyle can actually change the gut microbiome, and it can help in disease conditions. How we do it, I would not go into the details. We use metagenomic profiling for the benefit of students. 16S metagenomics and shotgun metagenomics are the two terms that you will hear. We use metagenomic profiling, taxonomy profiling to identify species. There are more than 500 to 1,000 different species in the gut microbiome. So we identified this using next generation sequencing or metagenomic profiling. How does this impact from a deceased individual to healthy individual? You'll see a lot of differences that we routinely see in the gut microbiome. So it is deceased individuals' microbiome is very different from a healthy individual's microbiome. Therefore, it can be easily used as a diagnostic tool for the health indicator or the health status of an individual. And diseases, when we say it's not just one, it is involved in gut microbiome is involved in liver disease, IBD, obesity, diabetes, cancer, infectious disease, psychological disease, autoimmune disease, everything. So gut microbiome, the role of microbiome to human health has been in every disease that we can think about. In India, this is more important as lifestyle diseases and non-communicable diseases kill about 61% of people. Our test called the Gut Microbiome Test is a profile of all the organisms present in your gut, and based on which we give diet and nutrition recommendations, prebiotic, probiotic recommendations, and your disease susceptibility to given diseases like diabetes, obesity, etc. This is how it looks like. We collect the sample, we do DNA extraction, we do sequencing in our lab in Mumbai, we do analysis, and some of the samples, when people ask, we do banking for future use, and we also give report and nutrition recommendations for individuals. So this is how our microbiome looks like. If you talk about not just gut, we are working constantly on all kinds of microbiome, like the oral microbiome, nasal microbiome, vaginal microbiome, skin microbiome. Everywhere microbes are resident parts of our body, and studying them and applying them to prevent disease and improve our health is very, very important and a constant endeavor in our company. Now I'll shift gears from infectious disease to wellness microbiome to cancer and genomics and cancer. Cancer has been the biggest beneficiary of all genomics applications. And cancer can happen in any of these tissues. This is a nice slide that talks about cancer can happen in the brain, lymphoid, breast, skin to colorectal, blood, everywhere. So that if that is the prevalence of cancer in every tissue, genome information is very, very important. This slide talks about that 4 lakh men and 4 lakh women died due to cancer in 2018 in India alone. 1 woman dies of cervical cancer every 8 minutes. These numbers are really very sad that this is how this disease has manifested in India and everywhere in the world. For every 2 women newly diagnosed with breast cancer, 1 woman dies of it. Tobacco and tobacco issue related cancer kills 3,500 people every day. But there is hope. There is hope that genomic data interventions can make better decisions for chemotherapy, surgery, radiation, and all targeted therapies. In addition, it can prevent cancer. And we in our company work on oral cancer, cervical cancer, breast cancer, lung cancer, and colorectal cancer. And we are happy to collaborate with researchers on any other cancer that comes up. How does it impact the patient? It identifies the tumor type and stage molecularly, accurately, precisely. It aids in chemotherapy and radiotherapy. It reveals important genetic mutations that drive carcinogenesis. It can be applied for targeted therapies which require genomic information. It can be used for optimization of clinical outcomes and avoidance of unnecessary therapies and minimizing the side effects. That is very, very important. This is how it is done. We do Illumina-based genome sequencing for, this is an example slide for 15 gene targets of pan cancer. So we identified 15 gene targets in one of the cancers, wherein the workflow goes like this. You have DNA, you have tissue first, you make DNA, you make library preparation, and you do the sequencing. And we found as low as 5% allele frequency. So in a given tissue, 5% of that tissue was mutated to cancer type. So that we were able to detect using Illumina whole genome sequencing assay. So that is the sensitivity of this technology, which is not available in any other technology. And it can be done from very low amounts of DNA. So that is the potential of this technology. This is how it is done. We have genes, we have mutations, and we report to the clinician that this gene is mutated in this particular location in this much percentage of cells. So that is the potential wherein clinician knows exactly what is happening at that time in a patient's situation. As I said in the beginning, bioinformatics or computation science is a big, big part of all genomics. And now it's going to be all biology related activities. So we have data, but how to analyze this data? And if there are computation or computer related students in the audience, I urge all of you to take up genomics. I mean, genomics, if you see, genomics is the real big data. We talk about e-commerce, etc. But if you see the data that is contained in human genome, for example, 3 GB of DNA is contained in one human. And we have a population of 130 crores or 1.3 billion people. Imagine the vast amount of data that comes out of genomics can be handled only by very talented software engineers and IT professionals that India has fortunately seen a talented pool of IT and software professionals. If those people take up genomics, I urge them to take up genomics. These problems can be solved better. So this is the potential of bioinformatics and genomics, wherein every data has to be analyzed using proper coding, proper softwares. And machine learning, as all of you hear in everyday life, machine learning has been applied to e-commerce, etc. But genomics, if you apply machine learning, like Clive Humby long back, a famous mathematician said, data is going to be the new oil. So that is the magnitude of data that arises in the public, private, and commercial databases in genomics is unprecedented, wherein you have raw data, we clean up the data using softwares, we do feature extraction, and we do training for patterns. Like if a South Indian genome has a different pattern of genes than a North Indian genome, we identify that, we make models, machine learning models, and evaluate them to give accurate results for research. So AI, artificial intelligence, and ML, machine learning, are promising to be the breakthrough for medicine and personalized medicine. In our company, we do all sorts of services for education, for research, for medical community, healthcare, agriculture, everything. And this is a snapshot of what we do in our company. Whole genome sequencing to metagenomics to animal sequencing to transcriptome sequencing to discoveries like circular RNA, small RNA, RNA immunoprecipitation, chromatin immunoprecipitation, everything that you can think of, we are able to do here using mainly Illumina and Oxford Nanopore sequencers that are available in our lab. We cater to all clinicians, diagnostic labs, hospitals, academic research, universities, students and teachers like you, pharma companies, corporates, and the government. We have been working with all of them to make an ecosystem for genomics for all. That has been our vision from day one. And we continue to do that. Genomics should be available to all at an affordable cost. And that is where I would end. Please reach out to us. Please reach out to me for any questions. I'm always available for young students. I really feel the promise of India lies in these young students, and we can go a long way by our intellectual power and our talented pool of young students in genomics and every area of science. Thank you.
Thank you so much, Dr. Gautham. And I understand the complexity of the concept of genomics and next-gen sequencing, everything, but I appreciate the efforts that you have taken, keeping in mind about the background of the audience to make it as simple as possible. I understand it's a very tough job to explain that in a simpler way, but you have done a marvelous job. Thank you. Thank you so much, Dr. Gautham. One key takeaway that I would like to share with all my teachers, colleagues who have joined here is that Dr. Gautham is a good example of the career choices one can make in life sciences. Please tell the students that being a doctor or being in the field of medicine or becoming a life science teacher is not the only option when they go for a BPC stream in their intermediate. Please encourage them to pursue science, and they have wonderful career in the industry. They can become the startups entrepreneurs tomorrow and build the nation. They have the capacity to do that, and please motivate them to look for alternate careers. And tomorrow we have more sessions. We have a session exclusively dedicated for careers in life sciences and networking. So please attend that session and also share that knowledge with the students. I once again thank Dr. Gautham, and thank you so much for explaining about the MyBiome Therapeutics too. Hopefully we can collaborate for some educational collaboration or academic collaboration with you. TSveries is a huge organization in the Telangana state with over 268 institutions. We have thousands of students. So we look forward to a positive collaboration with your company as well, Dr. Gautham. Thank you so much for accepting our invitation. Thanks again. I have an announcement to the participants. I really appreciate that you have been really patient with all the lectures, and you have been attending very well. We'll now break for lunch and we'll join again at 2 p.m. for the poster sessions. So please do join the poster sessions. We have interesting art displays as well from our students. So I'm sure you would be delighted to go through them and know more about what inspired them to create the art that they've made and submitted. See you all at lunch. Thank you so much. You can use the same meeting link. Thank you, Dr. Gautham, again.
Thank you. Thank you.
Yeah, bye.
Thank you, Dr. Gautham. It was fantastic.
So, yeah. Shall we stop the meeting? Jagdish, is it okay if we just close this meeting and restart it again post-lunch?
Sure, ma'am. It's also good for the recording. Otherwise, it will be recording all the time.
Yes, yes,
Also,
yes.
YouTube. Yeah.
Okay.
Let's close it.
Yeah. See you post-lunch, Jagdish. Yeah.
What time you are meeting? I think 1 hour from now, right? Yeah.
It's
okay,
2 o'clock. It's a very short lunch break.
Sure, ma'am. See you.
Yeah. Okay.